Chemiosmosis and oxidative phosphorylation. So, as the name would suggest here, we're talking about chemical gradient, chemiosmosis, and we're talking about phosphorylation, so adding phosphates to things. This is more commonly referred to as the electron transport chain, and that is the structures that are going to be involved in using a chemical gradient in order to phosphorylate ADP into ATP. This is where we use our electron carriers. So, where we are so far. Glycolysis is out in the cytosol, and we are taking our glucose and we are running it through 10 steps, and we're getting pyruvate out of it. We get a tiny bit of ATP out of that as well, but not much. We put in 2 and we get out 4, so we're getting 2 out there. This pyruvate, however, is then able to be used in the mitochondria to generate some more ATP in the Krebs cycle. So we can run it through, again, the pyruvate oxidation. We can run that through and get into our Krebs cycle, and we can produce some more ATP. What we have been talking about as well, though, is that we get these electron carriers generated as well during the glycolysis step, as well as pyruvate oxidation, as well as the Krebs cycle itself. We are producing electron carriers, and this is now where we're going to use those electron carriers. So far, we have not generated much ATP, but we have generated a little bit, and we can start putting our electron carriers into what's called the electron transport chain in order to oxidatively phosphorylate our ADPs into ATPs, and that's where we're going to get a bulk of our energy production during cellular respiration. So glycolysis, looking at the numbers, we got 2 ATPs out. Remember, we did make 4, but we took cost 2 to make that. We get 2 NADH electron carriers. Pyruvate oxidation gave us 2 more, as well as using up 2 of the carbons. Krebs cycle gave us 6 more NADHs plus our 2 FADHs, so more electron carriers. And again, that used up all of our carbons, and we've turned our entire glucose molecule matter-wise into carbon dioxide, and other things are going to stay in the system there. The ATP generation to this point, though, we have 2 ATPs during glycolysis, 2 ATPs, which again we had to make the GTPs first, and then we use them to make the ATPs. So we so far have 4 ATPs, which is not a lot. But again, these electron carriers are now generated, and they're all ready to be put into our electron transport chain to oxidatively phosphorylate some more ATPs using a chemical gradient. Energy-wise, this is the overall view of where we are so far. So in terms of the molecules we started off with, remember glucose, the first part of glycolysis, we're putting in energy. So you notice that we are actually putting in energy. This is an endothermic process. If you don't count in the ATPs, it's endergonic. Since the ATPs are there, they're able to couple it and get that to be overall exergonic. So it is possible to actually do the process itself only through the consumption of some ATPs. But we then are able to generate pyruvate and get out more ATPs than we put in. And so overall glycolysis is an exergonic process. It is giving us 2 extra ATPs, which is good. And if you're a bacteria, that's fine. You can make your living doing that. But what higher energy need organisms can do is they can continue on with this process to take your pyruvate and really reform it all the way down to oxaloacetate, which will then be reused in the cycle. And you can see this is definitely also exergonic. You're able to get a lot of energy out of that. And a lot of that energy is in the form of these electron carriers, a little bit of ATP, but essentially these electron carriers, they are all going to be used in the electron transport chain to get the remainder amount of energy from the original glucose molecule through several conversions, of course, but here's where we actually start making some energy. So electron transport chain is going to be taking these electron carriers, both the NADHs that we made and the FADHs, and we're going to be able to make not just 2 or 4 ATPs, but we're able to make up in the high 30s of ATPs. These are average numbers, so it's not going to go perfectly every time, but this is sort of the amount that you could expect. Remember, we're talking the mitochondria here, and so this is the inner membrane of the mitochondria is where these structures are going to be found. And the cristae, those when you go to draw a mitochondria, there are little bumps inside of it. Those are the cristae, the inner membrane, and in that inner membrane, there are these hydrogen ion pumps. And so they will be able to create a gradient of hydrogen ions. This is the chemiosmosis part. So we're able to create a gradient, and then the hydrogen ions are going to move down that gradient. Remember, a gradient would be creating an area of high concentration relative to an area of low concentration, and so they're going to want to naturally move back to even. And so once you make that gradient, they'll move back to even, and we can use that to do work. The work that we want to get done is make ATP. So we use the hydrogen ion gradient to phosphorylate our ADPs into ATPs. This is going to be requiring oxygen. So we've talked about our carbons so far. We've tracked our glucose and we've made our 6 carbons. That's good. But you'll notice that the chemical reaction for cellular respiration requires oxygen, as do you as you breathe. This is why we need it. We don't need it at all to break down the glucose. That's no problem. But if we want to use those electron carriers that we generated during that destruction of glucose, we now need the oxygen in order to utilize those electron carriers. The FADHs that we made in the Krebs cycle, we're going to get about 2 ATPs from each of those, and NADH is able to hold, think of it like a higher energy version of the electron. It's still holding electrons, but they're able to produce more ATPs per NADH used. The NADHs, however, that are made during glycolysis aren't in the mitochondria, so they've got to get there, and that can sometimes reduce the amount of energy that they have. It may have to go through a particular process that uses up some of that energy, and so that's why you'll see the variation in the amounts. Not only is it sort of an average between all of it, but again, the transport costs of those NADHs during glycolysis could decrease their amount. So we have our charged, our full electron carrier, and it can let go of its electrons and form back into NAD+. It's a non-carrying version. This hydrogen ion has an electron, so we have, as written here, we've got our hydrogen with its electron on it, and then it can be separated from its electron, forming a hydrogen ion itself. So you can think of it as the NADH that's holding that electron is also holding onto a hydrogen. So think the hydrogen is cleaved from the NADH or the FADH2, and the hydrogen itself is going to have its electrons removed from it, creating a hydrogen ion. So these electrons that are removed are going to pass along from one carrier to the next. So NADH and FADH2 are sort of the mobile carriers that move it from our cellular respiration processes into our electron transport chain. But the electron transport chain itself, the chain is made up of things that pass electrons from one to the other. This will allow the free energy, the ability to do work, to be converted into the pumping of hydrogen ions across the inner membrane into the intermembrane space. So think of the hydrogen ions being like in this matrix here, and they're going to get pumped into, not out of the mitochondria, but into the space that's between the 2 phospholipid bilayers. So this is the intermembrane space. It's the space between the 2 membranes. That's where we're pumping the hydrogen ions. So you can imagine a bunch of hydrogen ions inside. We can use the free energy of the electrons from the electron carriers to pump those hydrogen ions into this little gap that is between the 2 membranes. Just to look at the hydrogens, again, we tend to ignore them in their complexity, but if you think of the NAD plus picking up electrons, it does that while picking up a hydrogen, and so it forms this NADH, which is really a hydrogen plus the 2 electrons plus what it was originally, and then it can let go of those 2 electrons, and again, they can be used to drive the pumps, and what's being pumped are these hydrogen ions. This NAD plus can then go back to the beginning and be reused again during, let's say, glycolysis or Krebs cycle, pyruvate oxidation. So here's the schematic for the structures involved in it. The structures themselves have different names, and again, this is in the matrix here. This is where we have our NADH dropping off its electrons. The electrons are going to be following this arrow that we see through here. So again, they're moving from one electron carrier to the other, and as they do this, what's going to happen is the electrons themselves are moving, and that is allowing a conformation change in these proteins that pump hydrogen ions into the intermembrane space out of the matrix. So as these electrons are moving, we're getting hydrogen ions pumped into the intermembrane space. The electrons move down this chain of carriers, and each time using some of their free energy to pump hydrogen ions, and we end up forming a high concentration of hydrogen ions on the intermembrane side and a low, so this is the high concentration side, this is the low concentration side, and this creates our chemical gradient. At the end of the chain, we have this fellow here, oxygen. Oxygen, high electronegativity, it really likes electrons. So oxygen will easily gain electrons, and it is the destination, the final destination at the end of the electron transport chain. It is going to combine with some hydrogen ions to produce water. And that will also, so this is further lowers the concentration of hydrogen ions in the matrix, because not only are they being pumped out of the matrix into the intermembrane space, but they're also being consumed and turned into water with the addition of the electrons from the chain, the hydrogens and the oxygens all come together and form water. Same thing is going to be happening with FADH2. It happens a little further down the line, so we don't get quite as many hydrogen ions pumped, and therefore we don't get quite as many ATPs produced per FADH2, but the same process is going to happen. It's just going to not pump as many hydrogens and therefore not create as many ATPs. So NADH is oxidized, we lose 2 electrons, they travel down the electron transport chain. As they move down that chain, hydrogen ions are pumped, creating a gradient. This hydrogen ion gradient is what we use to make ATPs. The thing that does it is called, appropriately, ATP synthase. The FADHs do the same thing, but they don't pump as many hydrogens, and therefore we get fewer ATPs. Here's a slightly more complex model showing some more of the structure. So again, each structure has its name. The electrons are moving through them. The hydrogen ions are being pumped. So all of that is happening. This thing at the end here, this is what is going to use that hydrogen ion gradient. So it is able to essentially run on the high concentration of hydrogen ions in the intermembrane space, and it's ATP synthase. So we've got this complex sort of machine, enzyme machine, that is going to take ADP and turn it into ATP. So it's able to phosphorylate ADPs back into their charged version, ATP. Now, obviously, that would be an endergonic process. If you're charging up an ATP, you're putting that phosphate back on. That's going to require energy, and that energy comes from the high concentration of hydrogen ions in the intermembrane space. Schematically or graphically, we should probably look at as the electron transport chain starts with these high energy electrons, all electrons, but I think ability to do more work electrons. They're going to come in at the beginning of the chain, and they're going to be passed down through the pieces of the chain and eventually end up with oxygen in the form of water, which is sort of a low state of being. So they go from a high energy to a low energy. In doing so, they pump hydrogen ions, which then are used to generate, their gradient is used to generate ATP. So electrons move from one carrier to the other, sort of downhill in terms of their ability to do work, their free energy, and eventually they end up at oxygen. Each electron carrier is more electronegative than the last. This is why they're moving from one to the next to the next, or from one to the next to the next to the next. And eventually oxygen is going to be the most electronegative, so it is the one that they're trying to get to. This is the controlled release of that free energy contained within those electrons that were carried here by the electron carriers, the electron carriers themselves being produced during the previous steps of cellular respiration. Here is a more detailed version of ATP synthase. So again, think of it like a little machine that sits in the membrane here, the inner membrane, and these hydrogen ions, which remember previously they all got pumped to the intermembrane space, so now there's a high concentration of them here. They will move back down their concentration gradient, as things will always do, and as they do that, they will allow this machine here to take ADP, attach a phosphate onto it, and produce ATP. So the exergonic process of allowing this gradient to be equalized, allowing the hydrogen ions back down to an equal concentration, that exergonic process is coupled with the endergonic process of producing ATP. So once the hydrogen ion gradient is created, they're able to diffuse through this ATP synthase machine, and this is going to synthesize those ATPs, phosphorylate ADP into ATP. Summary of the entire process, one more time. So, glycolysis produces pyruvate. Pyruvate is turned into acetyl-CoA, and it produces electron carriers as it does that, as we had with our glycolysis also produces electron carriers, which is going to have to make its way into the membrane. More on that in a minute. Acetyl-CoA is then going to be used in the Krebs cycle. Again, we get these electron carriers generated. We get a little bit of ATP that can be used there, but these electron carriers that we have generated from these previous steps all make it to our electron transport chain. As they move through the chain, they eventually, make sure I go the right way, they eventually combine with oxygen and hydrogen ions to make water in the matrix here, further reducing the hydrogen ion concentration. But also as they move through the chain, they're pumping hydrogen ions out into the intermembrane space. So the electrons are harvested from the electron carriers into the electron transport chain. The electrons move through those various structures within the chain, pumping those hydrogen ions. Remember, another name for hydrogen ions is protons. So these are known as proton pumps. Eventually, once they've gone through all of those proton pumps, the electrons then end up with oxygen to form water. And we end up actually further decreasing the hydrogen ion concentration, which is the same thing as saying increasing the hydrogen ion concentration on the other side of the inner membrane. That concentration gradient is then used with ATP synthase. The hydrogen ions come back down into the inner membrane space here. I should say that wrong. They come back down into the matrix, and in doing so, this ATP synthase is able to take an ADP and turn it into ATP. 10 NADHs will lose their electrons. 2 FADHs will lose their electrons. So these are all the ones we've produced in the previous steps of cellular respiration. The oxygens that are needed, we will need 6 oxygens here. We do know that 6 overall per carbon dioxide, so it is a total of 6 oxygens. Tracking water properly throughout the entire cellular respiration is a bit of a job, and since it has hydrogen in it, we'll count it up in one of those things that we don't keep track of, but we are definitely going to be using up 6 oxygens for every glucose that we run this through. And once you work out all of the waters through all of the processes going in and out, as we saw, we're going to have many processes like Krebs cycles is producing water and consuming water. So once you get it all summed up, you would end up with 6 of those produced. Total 6, not just during electron transport chain. So that's a total of 6. And importantly for us, you will end up with specifically the electron transport chain, 32 to 34 ATPs produced in this particular process of cellular respiration. So here's the numbers. Glycolysis gives us 2 ATPs. It made 4, but it cost us 2. Gives us 2 NADHs, which can give us 4 or 6, because remember they're out in the cytosol, so sometimes it's tricky to get them in. So if we're lucky, we get 6. Otherwise, we'll only get 4. Once we're in the cytosol, the pyruvate can go through pyruvate oxidation, and our 2 NADHs that are generated during that process are definitely going to give us 6 ATPs. The Krebs cycle is going to give us directly, well, not really directly, it gives us 2 GTPs, which then get made into 2 ATPs, so we get 2 ATPs out of that. The 6 electron carriers, doing the same math we did before, will give us 18 ATPs. The NADH electron carriers, the FADH2 that we produced, the 2 that we make in the Krebs cycle will give us 4 ATPs. So that gives us a grand total of 36 ATPs per glucose molecule. And if we're lucky, we might get 2 more than that if we can get good shipping costs on the electron carriers into the matrix. But we can sort of count on 36 or 38 depending on how we get those electron carriers into the matrix. This is what's costing that transport into the matrix. So this NADH that we have out here in the outer membrane, we've got to get it into the matrix here. This could cost, depending on which sort of shuttle it uses. If it gets converted to FADH, we know that that FADH produces fewer ATPs. If we can get it through as NADH, we'll get more ATPs. So the question really is, those electron carriers during glycolysis, can we get them in directly, or do we need to turn them into FADHs? And that's where we get the variation between the 36 or the 38. This process of pumping electrons and allowing those hydrogen ion gradients to generate is great because we can use that to make ATPs. But sometimes those hydrogen ions can be allowed, those protons can be allowed to just run through the membrane without using ATP synthase. So we're essentially just allowing the gradient to sort of empty itself out. In this process, we actually have special proteins that can allow it to sort of short circuit the ATP synthase and sort of bypass it and go directly down back into the matrix. When this happens, heat is produced. Not unlike a electrical short circuit almost. So instead of running through ATP synthase, it runs through this other protein which doesn't really generate any ATP. It just allows the gradient to go back to, or closer back to equilibrium, and this makes heat. So these uncoupling proteins are specifically made so that we do actually purposely make them, which is kind of weird because you think, well, why wouldn't you want ATP? Why would you make something that short circuits the ATP synthase? But they are actually something that can be made. And if you think about it, not only do we need energy, but especially us as endotherms are sometimes going to need heat as well. So you can think of certain animals such as birds trying to stay warm in the winter, or hibernating bears, or even you when you had more surface area to volume ratio. You may need to generate a lot of heat in order to keep your body temperature at what it needs to be. So allowing this uncoupling of ATP synthase is one of those ways that our body does actually generate heat, and it does by having these special uncoupling proteins destroying the gradient instead of using it to make ATP, and in the process making heat, which sometimes is very valuable.